Welcome back to Chapter 6, Enzymes. In this lecture, we will discuss some of the examples of enzymatic reaction mechanisms. Chymotrypsin is a protease, which means that it can cleave peptide bonds. A protease is a protein that digests other proteins. And when I say digest, it means cleaving the peptide bonds in a protein and making individual peptides from them. Proteases are commonly found in the digestive tract as well as in cytoplasmic compartments. There are so many different proteases that can function at acidic pH in our gut. Most proteases recognize specific amino acids or amino acid signatures and cleave the peptide bonds linking these amino acids. If you remember, we looked at some proteases in chapter 3, and among those proteases, some of them happens to be peptidases. Shown on the right is the structure of chymotrypsin. The blue colored surface representation helps in understanding how a substrate binds in a pocket. For example, the red colored region that is shown here are the amino acids that are found in the active site of this enzyme. The yellow colored region is where the substrate binds. Chymotrypsin cleaves the peptide bond adjacent to aromatic amino acids. And these three are aromatic amino acids, right? Phenylalanine, tryptophan, and tyrosine. This enzyme, chymotrypsin, requires 4 critical amino acids to form the active site. And as you can see in this diagram here, this is a zoomed in version of this structure. There are 4 different amino acids: aspartate 102, histidine 57, serine 195, and glycine 193. These are the 4 amino acids that are responsible for catalyzing the cleavage of a peptide bond. And a substrate of this enzyme looks like this. AA could be any amino acid that is followed by an aromatic amino acid. An example that is shown here is phenylalanine. And chymotrypsin cleaves on the C terminus of this aromatic amino acid. And after cleaving, you can see there are 2 products, product 1 and product 2. In chymotrypsin, serine, histidine, and aspartate form a hydrogen bonding network, as shown here in this diagram. Aspartate hydrogen bonds with this histidine, which donates a hydrogen bond, and serine OH donates a hydrogen bond to the other nitrogen of histidine 57. The combination of serine, histidine, aspartate in a hydrogen bonding network facilitating catalysis is called as a catalytic triad. Interactions of the polar and ionizable side chains allows for changes in the pKa of this histidine 57. As you may know, histidine has a pKa of 6.5 for its side chain. Now that pKa can be tuned if you allow enough hydrogen bonding interactions between its nitrogen on the side chain. This facilitates the serine side chain to attack the target carbonyl carbon of the substrate. And it is this catalytic triad that is responsible for the mechanism of reaction of chymotrypsin. Let us consider the chymotrypsin mechanism. And shown here is a simplistic mechanism of this reaction. To understand the detailed mechanism, we'll have to refresh our organic chemistry knowledge. And to understand that, let's look at how one can draw chemical mechanisms. As you may know, a covalent bond consists of a shared pair of electrons. Non-bonded electrons important to the reaction mechanisms are designated by electron pairs, either as two dots or as minus. Curved arrows that shown here in red represent the movement of these electron pairs. For movement of a single electron pair, we usually show just a one hooked arrow. It's also called as a single-headed arrow. But we won't be using a single electron mechanisms. We will be using mechanisms involving electron pairs. You may also remember from your Chem 1 and Chem 2 lectures that some atoms are more electronegative than others. That is, they more strongly attract electrons. For example, oxygen and nitrogen are more electronegative than carbon. And that is precisely why when a carbon is bonded to an oxygen, the electron pair is attracted towards the oxygen. Now, let us look at some examples of nucleophiles and electrophiles. Nucleophiles are negatively charged and has an electron pair with them. For example, O- or an alkoxide ion is a negatively charged oxygen, as in an unprotonated hydroxyl group or an ionized carboxylic acid. S- is a negatively charged sulfhydryl group. When cysteines are deprotonated, you get S-. When serines are deprotonated, you get O-. Carbanion, C-, is a nucleophile. Uncharged amine groups can also act as nucleophiles. Imidazole, which is the side chain of histidines, can act as nucleophiles. Hydroxide ion, just hydroxide ion, can also act as a nucleophile. Now let us consider electrophiles. A carbonyl group acts as an electrophile. The carbon of the carbonyl group is electron deficient. And the reason is because the more electronegative oxygen pulls the electron pair towards itself. And as a result, there is a net positive charge at this carbon. And hence, it is electron deficient and is an electrophile. As you can see, the arrow is pushed towards the electrophile and it starts from a nucleophile. Protonated imine group also is an electrophile. The carbon of the imine group is electron deficient and is activated for nucleophilic attack at this center. And hence, the arrow is pushed towards it. And once you push the arrow towards the carbon, you push the electron pairs into the heteroatom. And in this case, the nitrogen. In the previous case, it was the oxygen. Phosphorus of a phosphate group is also an electrophile. A nucleophile attacks the phosphorus center and the electron pair is pushed towards the oxygen. A proton is also an electrophile. A nucleophile can attack a proton. Step 1 of chymotrypsin reaction involves binding of a substrate. this is E and this is S, right? E when encounters S or substrate forms the ES complex, as we have seen before. The active site is decorated by 4 amino acids, as we saw before. Glycine, backbone of glycine 193, that is the NH, and the backbone of serine 195, the NH. They form what is called as an oxyanion hole. And I'll explain as to what this oxyanion hole is in a bit. At the same time, the side chain of serine hydrogen bonds with histidine, which in turn hydrogen bonds with aspartate. Now this enzyme is ready to bind a substrate, and a substrate in the form of a polypeptide binds in this specific region of the enzyme. And once it binds, all the side chains of this peptide, they fall in the right place. For example, this hydrophobic pocket is occupied by this hydrophobic amino acid, which in this case is a phenylalanine. And this allows the enzyme to juxtapose this specific peptide right next to the catalytic triad so that it can catalyze the peptide cleavage reaction. Once the substrate binds, the lone pair of histidine 57 abstracts the proton from the side chain of serine 195, and as a result, a strong nucleophilic alkoxide ion forms on the side chain of serine 195. And this alkoxide ion then initiates a nucleophilic attack on the carbonyl carbon of the substrate, and this attacks the specific carbonyl carbon of the C terminus of phenylalanine, as shown here. Now this is accompanied by the formation of a short-lived negative charge on the carbonyl carbon. Now this negative charge on the carbonyl carbon is called as the oxyanion, and that oxyanion is stabilized by the backbone NH from glycine 193 and serine 195. And if you don't stabilize this oxyanion, the enzyme cannot stabilize the transition state. Now, this oxyanion is, because it's short-lived, it gets stabilized by the oxyanion hole. And once it's stabilized, the oxyanion, the oxygen, the electron pair on the oxygen pushes back and pushes out the peptide that is on the C terminus of phenylalanine. And that peptide leaves with a proton that is attached to histidine 57. This slide reiterates what I just said in the previous slide. The oxyanion on this carbonyl oxygen is stabilized by the backbone NH from glycine and serine. Now, because of this instability of this oxyanion, this tetrahedral kind of an intermediate collapses. And once it collapses, the peptide chain attached to the C terminus of phenylalanine leaves, and the leaving group is protonated by histidine 57, which facilitates its displacement. As a result, the enzyme is now stuck with an acyl group, and this enzyme substrate intermediate is termed as an acyl enzyme intermediate. This covalent acyl enzyme intermediate that is shown here is a very short-lived intermediate, and there is sufficient evidence from kinetics and also from mass spectrometric studies suggesting that acyl enzyme intermediate is indeed short-lived and can be observed. Once this acyl enzyme intermediate forms, a water in the active site which gets deprotonated by a histidine residue, the same histidine 57. As a result, an alkoxide or a hydroxide ion is formed. This hydroxide attacks the carbonyl carbon of the acyl enzyme intermediate. As a result, electron pairs get pushed into the carbonyl oxygen, which is now an oxyanion. And this second tetrahedral intermediate is again stabilized by the oxyanion hole. The second oxyanion is also stabilized by the hydrogen bonds at the oxyanion hole. Now, again, because this oxyanion hole is unstable, it collapses, and as a result, the electron is pushed out, and it pushes out the serine, and serine now abstracts a proton from histidine 57. Again, this is the same diagram from the previous slide, and as a result of the collapse of the tetrahedral intermediate, you're left with the product. Now, this is the second product, and once this product is released from the enzyme, you will get product 2, and this is enzyme product complex 2, so you could say EP. The product has not been released yet. Product dissociation or product release is the final step in this chymotrypsin mechanism. Product dissociation results in the active enzyme. So this step regenerates the free enzyme, and this goes back for another round of peptide hydrolysis. Next, let's consider a reaction that is opposite of peptide bond hydrolysis, and this is peptide bond formation. Transpeptidases are a group of enzymes in bacteria that cross-links peptides in peptidoglycan of bacteria. For example, Staph aureus is a gram-positive bacteria that has this kind of a peptide structure in its peptidoglycan. As you can see, there are five amino acids in this peptide: L-alanine, D-glutamate, L-lysine, D-alanine, and D-alanine. Transpeptidase, an enzyme, uses a serine residue in its active site, and it first forms an acyl enzyme intermediate by attacking this carbonyl carbon of the penultimate D-alanine in this peptide. This results in the release of the D-alanine, the terminal D-alanine, resulting in the formation of an acyl enzyme intermediate. Now, this acyl enzyme intermediate is then attacked by